Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is part of our Biochemistry Focus webinar series. Topics in the series include research highlights from many areas of the molecular biosciences and also practical sessions to support career development. Each webinar gives you an opportunity to ask questions by text. We also welcome suggestions for future topics and speakers to feature in our webinar series. Please see the webinar for more details. I am Patricia Kuwabara, professor at the University of Bristol and a trustee of the Royal Society of Biology. The mission of the Royal Society of Biology, or RSB, is to be the unifying voice for biology, to facilitate the promotion of new discoveries in biological science for national and international benefit, and to engage the wider public with our work. I would like to highlight that the Royal Society of Biology is currently celebrating Biology Week by showcasing the amazing world of the biosciences through a number of events and activities, which are all virtual this week due to the pandemic. For more information about activities and membership, please check the RSB website at rsb.org.uk. So, on this note, it is my distinct privilege to chair this afternoon's webinar. The webinar features an exciting set of talks addressing new research developments and directions being taken to address global issues affecting human disease and healthcare. Today's webinar is entitled, "A Decade in Biology: What Have the Biosciences Done for Us?" It is linked to the special issue of the journal, Emerging Topics in Life Science, published jointly with the Biochemical Society, Portland Press, and the Royal Society of Biology, celebrating the last 10 years of the Royal Society of Biology. We will now listen to Professor Richard Rees tell you more about this special issue.
Hello, my name is Richard Rees. I'm Professor of Molecular Biology and Deputy Vice-Chancellor at the University of Kent. In addition, I'm the Honorary Secretary of the Royal Society of Biology. I also had the enormous pleasure and privilege of guest editing the special issue of Emerging Topics in Life Sciences with a sub-theme of celebrating 10 years of the Royal Society of Biology. The biosciences have never been more important to both understanding and addressing global health, wealth, and well-being. In the 10 years since the formation of the RSB, we have seen enormous changes in the pace of biological research and the ways in which this research impacts our everyday lives. Emerging Topics in Life Sciences is a journal published jointly by the Biochemical Society, Portland Press, and the Royal Society of Biology. As part of the Biology Week celebrations, we are providing free access to the latest issue until October the 11th. Today, we welcome a series of talks from different speakers who have contributed to the special issue and will cover topics including looking at various aspects of human health. You could read more about each of these areas and much more by going to the Emerging Topics in Life Sciences website. I hope that you enjoy the presentations. Thank you.
In this webinar, we welcome 3 speakers who kindly contributed papers to this special issue and who are now providing talks showcasing what the biosciences have done for us. Unfortunately, our 4th invited speaker, Dr. James Cherry from UCLA, won't be able to join us today, but he will be presenting his talk, The Sociology of the Anti-Vaccine Movement, in one of our upcoming webinars on Wednesday, the 2nd of December at 1700 GMT. Registration will soon be available, so please keep an eye on the Biochemical Society's website at www.biochemistry.org/webinar for more details. His presentation is guaranteed to garner great interest in view of the current decline in the coverage of childhood vaccinations, and also because fast-track vaccine development has taken center stage in the race to find therapeutics to contain the COVID-19 pandemic. Before I introduce our first speaker, please be aware that there will be a question and answer session at the end of each presentation. I encourage you to send in your questions during the talk. When you have a question, please type it in the question box as shown in the image on the screen. So without further ado, our first invited speaker today is Dr. Karl Frontzek. He is a consultant neuropathologist and senior researcher at the Institute of Neuropathology, University of Zurich, Switzerland. His scientific work is focused on immunological aspects of prion diseases. Today, he will be presenting his talk, Recent Developments in Antibody Therapeutics Against Prion Disease. Dr. Frontzek, the floor is yours.
Patty, thank you very much for the kind introduction.
Your screen is not yet up. There you go.
Okay. Yes, so hello everybody, and I'm glad that you attended this seminar. I'm happy to tell you something about a topic that I have been researching for the last years, which I will now summarize in the presentation called Recent Developments in Antibody Therapeutics Against Prion Diseases. As Patty said, I'm a consultant neuropathologist at the Swiss National Reference Center of Prion Diseases. So we handle the humans that suffer from prion diseases in Switzerland. And we also do research on prion diseases. To disclose, I received industry funding from UNO Pharmaceuticals and public funding from University of Zurich and other Swiss and American public foundations. I would like to introduce you to prion diseases. Prion diseases are rare diseases. They occur in humans with an incidence of about 1 or 2 patients per million inhabitants. Prion diseases are also called transmissible spongiform encephalopathies. This is because when you look at the brains of patients succumbing to prion diseases, the brain has the very characteristic so-called spongiform or sponge-like histology. As you can see here, this is an histological picture of a patient with prion diseases. There is widespread vacuolation, but also neuronal loss and reactive gliosis of the central nervous system. Today, we believe that the cause of prion diseases is due to misfolding of the cellular prion protein, which is ubiquitously expressed in the brain and also extraneural organs in humans. By mechanisms that are not yet known, it transforms towards a pathological aggregated conformer, which is called the disease-causing prion, or also the scrapie prion. Today, prion diseases are still inevitably fatal diseases, and there is no cure yet. So the prion diseases have gained strong momentum, especially the research on prion diseases during the mad cow zoonotic, which happened mostly in the UK and also other countries in Europe and worldwide, by transmission of probably prion-infected cattle to humans. Since then, there have been many approaches, mostly preclinical, to treat prion diseases, starting with antibody therapy, which is aimed at stopping the conversion from the physiological to pathological protein, but also to compounds targeting prion replication, and eventually halting intracellular pathology, such as enhanced degradation of pathological proteins, as well as chaperone therapy for refolding of misfolded proteins and others. So today, my talk will be focused on antibody therapy, as this also currently is one of the 2 therapeutic modalities where clinical trials are ongoing. So to give you a brief recap about what therapies have been already tried against prion diseases. In the last 20 years, there were a total of three controlled trials. So the furthest advanced was the doxycycline trial. Doxycycline is actually an antibiotic. However, it was found that doxycycline potently ablates prion infectivity. So Haig and al. Performed a phase 2 study on patients mostly with sporadic Creutzfeldt-Jakob's disease. So this is the most common prion disease in humans. However, after the first interim analysis, the trial was cancelled because there was no benefit observable. Another drug, which is called flupertine, this is an analgesic, was tested by Otto et al. In a similar setting as Haig's study. While Otto and al. Saw that there was a better cognitive function of patients receiving flupertine, the survival times were mostly unchanged. Lastly, quinacrine, which is an antimalarial, was tested by Michael Gishwan's group and collaborators. However, comparing to the results from Otto et al., they also saw a slightly better cognitive function, but the survival times were unchanged. So in the last years, there was a paucity of trials against prion diseases. However, there is currently 2 trials which are ongoing, one of which is finished at the beginning of this year, namely an UK-based trial at the University College of London. This is PRN 100, which is a humanized anti-PRPC antibody, meaning that it targets the physiological prion protein. This is a compassionate use trial in prion diseases, which was finished, but the results are still undisclosed. And more recently, this is from a group from the Broad Institute in the USA. This is a collaboration with Ionis Pharmaceuticals, led by Eric Minikel and Sonja Wallab, where the preclinical stage showed evidence of effectivity of antisense oligonucleotides against the prion protein gene. So after having introduced you to the recent developments in overall therapeutic efforts, I want to now focus on recent results from the literature on anti-PRPC antibody therapy. The effectiveness of therapeutic antibody that happened in the last years. It was already discovered by Ruth Gabinson, who was working in the group of Stanley Prusiner, who then went on to get a Nobel Prize for this discovery of prions. So what Ruth Gabinson actually did is she purified the scrapie prion protein, which was back then obtained by digesting the pathological brains with the proteinase. And then she administered this purified protein to hamsters, and this antiserum then, when given
Hello, it seems we've lost our sound from the
see here. I'm sorry?
We lost your sound temporarily.
Oh, is it better now?
Yes.
Hello? Oh, apologies. So, as I said, what they could see is on the right side, you can see the control antibody. And this is the gray color depicts the pathological prion protein, and the administration of the therapeutic antibody showed a reduction in the prion aggregates, and also the mice were prevented from developing the disease. So what is then the mechanism of those antibodies? And actually, this was a study from Adriano Guzzi as well, where Tiziano Sonati and co-workers tested a battery of antibodies against the prion protein. Here is the schematic of those antibodies. And you can appreciate that the left part is the N-terminus of the prion protein, which is unstructured. And there is a C-terminal, more globular part. And actually, what Tiziana saw was that antibodies that are against the C-terminal globular part, which is seen here, for instance, POM1, the C-terminal antibody, wreaks havoc in the brain of mice, while antibodies that are directed against the unstructured domain are not only innocuous, but also, which was then shown by another postdoc from this group, Uli Hermann, they were also protective against prion diseases. So then, when I came to the lab, what we did, we engineered actually the first bispecific antibody for prion disease therapy, and we linked the protective POM2 antibody with the toxic POM1 antibody. And interestingly, by blocking both epitopes, we could also see a potent rescue of the prion pathology. And actually, this is, to our knowledge, the first antibody that was given when prion pathology was already evident and was still protective. So what we then asked was, there is there is a rare form of prion disease, which is caused by genetic mutations. And two examples are depicted here, which is Gerstmann-Straussler-Scheinker's disease, and the other one is fatal familial insomnia. And interestingly, those diseases, they only manifest in high age, but these patients have the mutation already at birth or even before. So we were thinking, if there are protective antibodies, maybe those patients have protective antibodies in the blood that keep the disease on hold until high age, when there is too much of the pathological protein. But we were not successful, and we did not find any elevated antibodies in patients with genetic prion diseases when we compared them to their family members which did not have the mutation. But then we're wondering, so if there are antibodies, which we saw in the genetic prion disease patients, maybe if we look at the whole population, then somebody that would have these toxic antibodies, this person might suffer from a very severe neurological syndrome. So with Susie Senatore and Mark Emmenegger, we screened over 40,000 patients from the University Hospital here in Zurich, and we plotted then the reactivity of the antibody, of the prion antibodies. And we could see that there was actually a small subset, which is all the black dots you see here, that have those antiprion antibodies. And when we looked at the age of these patients, we could see that they are, it's more the older patients than the younger who have these antibodies. So, however, when we looked at the clinical histories, we did not, we were unsuccessful in doing a link between a disease, suggesting that these antibodies are most likely innocuous. So, and this will be my last slide, what is now upcoming on the prion disease antibody side? This is unpublished data from our group, which is currently in revision. So we have the crystal structure of the toxic prion antibody when it is bound to prion protein. And the interesting finding is that there is an H bond between these two alpha helices, which is this arginine, and a histidine, which is in close proximity to another alpha helix. However, when there is no POM1 bound, there is no H bond. So then we mutated each of the binding residues from this toxic POM1 antibody and turned it to an alanine. And interestingly, in two antibodies, which is the Y57A and the Y104A, we could see that those toxic antibodies are not only innocuous, but also they're protective against prion diseases. And when we did molecular dynamic simulation and also NMR spectroscopy, we could see that the presence of this H bond correlates positively with the toxicity of antibodies. So lastly, when we designed phages that specifically detected the free conformation, but not this toxic conformation, we also came up with antibodies that were also protective. And as you can see by the overlap, they seem to dock the similar epitope as the toxic antibody. So just to sum up, we think that the toxic antibodies and the real prions share a common site on the C-terminus of the prion protein, by which they might exert the toxicity. So with this, I would like to thank all of the collaborators, which is mostly at the University of Zurich, my PI, Adriano Guzzi, and our collaborators at the Institute of Biomedicine, Dr. Luca Barani, and I'm happy to take questions.
Thank you very much for that thoroughly engaging presentation. I think as we age, we all harbor some fear of developing any form of neurodegenerative condition. So your presentation has kind of introduced a hopeful ray of light in the treatment of these human prion diseases. So are you ready to take some questions?
Yes, of course.
Okay. So could I start by asking you, you were talking about the protective aspects of these perhaps autoantibodies to PRPC. Right. Are there any large-scale longitudinal studies in progress to look at this question in more detail? And what kind of, from what you're learning now, would you be looking at certain types of antibodies focusing on this region that you've determined to be perhaps neuroprotective?
Right, it's a good question. So actually, there is not a longitudinal study ongoing per se, but I can tell you what we also did. So we mined the repertoires of publicly available immunoglobulin sequencing, and actually we saw that there is the sequences are quite similar to our protective antibodies. So we have the impression that the at least in as a subset of individuals, there is the anti-prion antibodies that are protective. And also what I can tell you, what we're looking at is when we designed these protective antibodies, we think that this H latch where the H bond is formed, this would be a good target to develop a small molecule. So we're now planning to do small molecule screens to specifically inhibit this, this structural change.
So are these antibodies then recognizing conformational elements more than sequence elements? Right. Yes. So actually, the first protective antibodies, where I told you there was an epitope dependency, meaning the N-terminal were those that are protective. There it's linear epitopes. So, and the more recent discovered are conformational epitopes. And actually the one antibody which has been given to humans now is also a conformational epitope to a similar region of our protective antibody. I think maybe I have one last question for you. Can you comment on the delivery mode of getting these antibodies into the brain, or how you would deliver? Right. So actually, I mean, of course, there is the usual route, which is intravenous injection, which, as you may know, takes a lot of amount of antibodies. But something that I have not shown, and what we have, it actually looks quite promising. The mice are still healthy now. We have constructed brain-specific adreno-associated viruses. So we give adeno-associated viruses in the vein, but the route of action is only in the brain.
And we gave them now to prion-infected mice, and we really hope that they survive at least 1 to 2 weeks longer, and then we might see actually protection against prions.
Well, thank you very much. We'll keep our fingers crossed with your research going forward. So thank you for the questions. I think we're going to move on now to our next speaker. Our next invited speaker is Dr. Kim Hardie. She is an associate professor in the School of Life Sciences at the University of Nottingham, where her research group aims to decipher the complexity of polymicrobial biofilms, especially in the context of serious chronic infections and antimicrobial effectiveness. In collaboration, her group uses novel optical nanosensors and cutting-edge technological platforms such as cryo-orbisims to monitor environmental barriers to antimicrobial penetration using realistic infection models, and couples this with molecular microbiology to unravel the mechanisms in order to find novel ways to overcome AMR and to identify potential new antimicrobial targets. Today, she will be presenting her talk, Antibiotic Resistance. Dr. Hardie, I hand over to you.
Thank you. Sorry, I'm waiting for the slides to be shared.
Yeah, we're coming up.
Okay.
I believe you need to accept the webcam request.
Yeah, I can't see it at the moment.
Could be hidden behind your screen.
Oops. Oh, here it is.
There we go. You're ready to go.
Okay, so thank you very much for the opportunity to speak today about the fight against antimicrobial resistance in the last decade. I'm based at the University of Nottingham, as you said, and I've got some funding that was able to support some of the work presented here from the EU and also from Unilever. So antimicrobials are very good, as you can see in this video here. So they really work very well. So here we've got a bacteria which has got a green fluorescent protein put inside it, and so that's why it's showing the green light. And here you can see the antimicrobials being added and the lights going off. And they're so effective that they have come to underpin a number of routine surgeries and treatments for things like cancer. You can see in the building blocks building up here, and that we rely on them so much now that if they don't work anymore, then we really are in trouble, and there's lots of things that are going to have an effect on our well-being. And one of those would be the development of antimicrobial resistance. So there are a number of different antimicrobials that have been discovered, many of them at the beginning, just after the first one, penicillin, was found, and they represent a whole bunch of different chemistries. They also work in a number of different ways. So some of them don't enter the cell, they just attack the outside and make the cell weak or even burst them open. And others get inside and they can stop the genetic material being reproduced, or they can interfere with the production of the protein building blocks or the metabolism of the cell, and therefore the cells begin to die. Because they're so effective, we use an awful lot of them. So here's the tonnage usage over all sectors, and these figures are from 2016. And you can see that very high levels in the States and Spain, which reflects the large use in animal agriculture at the time there. 10 years ago, this was such a big problem, recognised, that it was predicted to be the next threat on a par with global warming. And the reasons for this is because once antibiotics come into the clinic, it's not very long afterwards that you see some antimicrobial resistance that is preventing them from being as effective as they could be. And the real wake-up call came in 2017 when a patient died from Klebsiella pneumonia that was resistant to every available antibiotic in the US. And you can see the more recent ones that have been introduced have got a very short lag time before this resistance appears. So it's becoming a really big problem. But this antimicrobial resistance isn't just spontaneously appearing in the clinic. It actually was already present, but in the soil, for example, where you have lots of different microbes. And it's here, it's suggested that it's used as competition against other microbes, or perhaps at low level production, even perhaps as a signaling molecule between antimicrobials. But because you have a mixed population of cells that are in red resistant to antimicrobials and in green sensitive, if the level of the antibiotic rises, then the sensitive ones will die off, leaving the resistant ones, which can take over the environment, multiplying and having a selective advantage. With them, so many of them around, they might come in contact with other microbes, and this antimicrobial resistance can be transferred between microbes. And this is usually done by the transfer of genetic material from one bacteria to another. And this becomes our real problem when it starts to happen within the clinic. Because we've got the variation in the function of the antimicrobials, we also have a lot of different variation in the way that the resistant mechanisms work. Some of them prevent the entry of antimicrobials, or if they get in, they pump them out. Others affect the actual target of the antimicrobial or inactivate it. They can change the target so that it no longer fits the antimicrobial. They could also produce a different version of the step so that the antimicrobial inhibited process is bypassed, or it could involve overproduction of the target, which dilutes the effect of the antimicrobial. So we used to think that this was really fast spread. So the beta-lactamase that was discovered in New Delhi spread throughout the world in less than a decade. But now we know that pandemics can spread even faster. So maybe that's not as scary as we thought, but it's still pretty scary if a new one came along, because it would have such a big impact on us all. But this sparked a review in 2016, a report by Jim O'Neill, which used the current figures of 700,000 deaths per year from antimicrobial resistance to predict what would happen if this multiplied across the globe. And in the end, it predicted there would be 10 million antimicrobial resistance causing deaths globally per year, which would have a very high financial as well as well-being impact on us all. So the O'Neill report had 10 recommendations, and since that came out 4 years ago, there has been some movement. So there is some positive change in that numerous international AMR awareness initiatives have been launched, and there's a reduced use of antimicrobials in agriculture and dentistry, which is helping to keep down the emergence of the antimicrobial resistance in the clinic. And everybody was becoming a little bit more aware about hygiene. But what was remaining at this point was the need to incentivize the research and transform it so that it was more effective to find new antimicrobials and to understand the antimicrobial resistance. And the reason that you need incentives to develop more antimicrobials is because it costs a lot of money to generate one, do the research, isolate it, purify it and produce it, and yet it would be kept on the back bench on the top shelf for use in dire circumstances when everything else has already been exhausted. There's also an issue with low to middle income countries, because in these countries the antibiotic prescription is not tightly regulated, so people can easily get hold of antibiotics, and so there's a lot more in circulation. And also there's reduced sanitation and hygiene in these places as well, and maybe even linked into the refugee crisis, the moving of people around the globe. that means that more antimicrobial, that there's less fighting against the infectious diseases, and therefore there's more need for the use of antimicrobials. And whilst there's more hygiene awareness, there's a need to get that actually embedded into real-time behavioural change. And alongside that, there was recognised a need to keep surveying the use of antimicrobials and the spread of AMR so that it can be counteracted, and to develop rapid diagnostics, because then that means that if you can quickly diagnose that you've got a bacterial infection, and you can quickly link that to its antimicrobial sensitivity, you can then target the use of the antimicrobials where it's really needed. And in the UK, a strategy in the beginning of 2019 was brought in that addressed some of these things by setting targets for the reduction of drug-resistant infections, although it didn't make completely clear how this surveillance was going to be monitored and which drugs it was actually going to be involving. There was also quite a lot of progress in making goals to reduce the amount of use of the antimicrobials in humans and also in animals, but it was not made clear how this would integrate with changes that would come about post-Brexit. And to target the need for money in the development of the antimicrobials, it was proposed that a new payment model would be brought in, and there was some suggestion that it might be of a life insurance model kind of payment, where instead of thinking of the price of the antimicrobials being linked to the volumes that would be used, you would think of the price being linked to how much damage would occur if they weren't used, to make them more viable. So there's clearly more to do. They can't just rely on the strategy, and there are some movements. For example, a new hygiene improvement policy was brought out in the UK, and this was to make sure that there was standardised practice brought in again across all the healthcare staff, and to support them in their common understanding of the importance of this. And there's a lot of buy-in to tackle antimicrobial resistance. So there's citizen science and sharing of resources, and also ways to show that you're in favour of action on this side. And back in 2019, we were thinking that the next greatest battle in our fight against antimicrobial resistance would be the realisation that most of the bacteria don't actually live free living in liquid, but they actually come together to form complex communities on surfaces. And these surfaces really vary. They're in the environment, such as in pipes or in a domestic washing machine, on your teeth, on you, and on indwelling devices. And the problem with these communities is they have bacteria embedded in a matrix, and the antimicrobials can't penetrate very well through this matrix, which means they can't reach the antimicrobials in the middle. There's also altered environments in this matrix, which might change the way that the antimicrobial work, and the bacteria within them are exposed to stresses and may even develop into a dormant, persistent stage when they wouldn't actually succumb to the effect of the antimicrobial. So understanding about biofilms was very important. And here we see some of our data where we've compared the sensitivity to free living bacteria to inhibit their growth in blue for different bacteria, both pathogens that are not so good and commensals that are quite friendly. And that dose is quite low compared to the amount that you would need to kill these bacteria, but that pales into the insignificance really against the amount of antimicrobial, in this case ciprofloxacin, that would be needed to kill the bacteria when they're in the biofilm. So the reason, as I said, was to do with the penetration of the antimicrobials through the biofilm microcolonies. And you can see here a fluorescent microscopy image where you've got dead bacteria on the outside of a microcolony which have been hit by the antimicrobial, but still living ones in the centre, which is what gives the selective conditions for antimicrobial resistance. In addition, because biofilm communities can survive for a long time, they will get exposed to more than one type of antimicrobial, and there can be combination effects. So, for example, this biocide triclosan is in many household products, including the washing that's used to decolonize before surgery. And the issue with this is that it builds up in the body, and it can be seen that while ciprofloxacin can really kill this bacteria here very quickly, and you can see that visually on a petri dish there, if the bacteria are pre-treated with triclosan, they survive, and you can't see any difference in that number there of colonies that are growing. So that shows that the combination effect is something that we have to consider in our treatment with antimicrobials in the future to prevent the development of resistance or tolerance against them. And so back then there was lots of research ongoing into new antimicrobials, looking to understand the biofilm architecture, the niches of the environment, and so to develop new antimicrobials that work much better. To change the surfaces on which they're growing as well, so that they don't grow. So, for example, the indwelling devices like catheters can be coated with polymers, or their topology can be changed so that biofilms no longer form on them. And what we need to know is how these biofilms and their antimicrobial resistance correlates with the clinical outcome, and also how to diagnose where the biofilms are, because you don't want to treat with a different strategy if there's actually no biofilm there. And it's becoming more and more evident that more than one microbe might be present in a biofilm, and that one can influence the resistance to antimicrobials of another organism. And then something else happened. Along came a pandemic, and this really made us think about whether the antimicrobial resistance was still an issue or not. But back in April, Laurie Burrows presented this really nice figure where she took the number of deaths from COVID-19 at the time, compared them to the annual death from flu, cancer, and the predicted 2050 death from AMR as well. And you can see it pales into insignificance. And taking the value of the number of deaths yesterday into consideration, okay, we're a bit more than we were for flu there, but we're still a very small amount compared to the predicted amount of deaths from ANR. So the next question becomes, how is the pandemic affecting the number of deaths from AMR? And there's some reports that it's actually making it worse, because there are more use of the antibiotics, so therefore more selective pressure for antimicrobial resistance, based on an unknown cure for the COVID, and also the reports, for example, this one that's coming out of the media, that came out in the media, that antibiotics might be effective. In addition to that, there are a number of cases of severe secondary bacterial infections that needed to be treated, and because they were in such ill patients, you'd get empirical treatment, not necessarily based on sensitivity of the secondary infection, which means that the antibiotic isn't being used where it might be most needed. And on top of that, because many of the initial consultations with GPs are via the telephone, they err on the side of caution to prescribe the antibiotic, which means that more will be being used where they're not really needed. There's also an issue that will affect the treatment with antibiotics, which might not be initially apparent, and that is that many of the countries severely affected by the pandemic are the major suppliers of the antibiotics. And then there was proposed to be a number of other knock-on effects. For example, the activity towards the pandemic means there's less surveillance on AMR, delays in registration, withdrawal from WHO support, low public awareness of the AMR, and also fatigue for the crisis. But it's not all bad news. Also proposed is that there are a number of opportunities to fight AMR buried within the the onslaught of the pandemic. So there will be some money going into infectious diseases that could be used for surveillance. There will be new diagnostics because they're really needed. There's also the awareness that we need to think about transmission from the environment or from animals to the human chain. Different legislations are going through. And also there's been a lot of public information campaigns which are going to help in raising the awareness of hygiene measures. And for example, as far as hand hygiene is involved, I'm sure you're all aware of the government announcements, social media, celebratory demonstrations, the normal media, and publications that give you the full information you need to do your hand hygiene really well and when you need to do it. So there is also hope because there's been lots of research. So over the last decade, the amount of publications in AMR has doubled. And this has been a result partly of using state-of-the-art methodologies and multidisciplinary science. And much of it's been shared freely online, which helps it to be disseminated. And there has also been a report about how beneficial it is to incorporate creative disciplines like art and architecture into the fight against AMR. So that might be another way forward. The other ways forward will be on new treatments, new antimicrobials. And we might be able to find these by looking through old drugs that have gone through all the trials, and so therefore they're safe, but then they may be not as effective against the syndrome that they were designed against, and repurpose them for killing antimicrobials. There may be some clues in ancient remedies, like medieval remedies, that we might be able to reinvent. And we might be able to get through these different compounds by using artificial intelligence as it's developed. And one study has already found a useful antimicrobial by using AI to go through more than 100 million different compounds. There are different strategies as well. I mentioned how polymicrobial communities have an effect on each other. So we might be able to harness our friendly microbiome to fight against pathogens, and also to target processes in the pathogens that are not essential, that will not generate so much resistance, like to have an antivirulence factor molecule to combat the microbes. And then we might want to actually inject something into the microbe rather than use chemicals. For example, the viruses that affect microbes might be able to be deployed to reduce the amount of microbes in an infection, like a wound, for bacteriophages. Or we might want to actually develop new vaccines so that we can inject ourselves and have the prevention rather than cure. But overall, it seems that to be able to fully overcome AMR, we need to work globally and collaboratively in the way that we're learning through the pandemic, because the low to middle income countries face significant and specific changes that require solutions before we're going to be fully effective. And we need to build on what we know from the pandemic to embrace the One Health dimension and think about the environment and animals as the source of the antimicrobials and the antimicrobial resistance that is a problem to us. Finally, I'd like to thank the people in my lab. So I highlighted the people who'd done some of the data generation that I looked at before. And with that, I'd like to thank you all for listening.
Thank you very much, Kim. That was an excellent summary of the AMR problem and the strategies that are being taken to fight this problem. So before we go to the question and answer question period, could I ask you to unmute yourself to show your webcam? Great, now we can see you. Thank you. Okay, so the first question I have for you is in regards to AMR, what does it mean for our current situation with COVID? Are we using too much sanitizer or antibiotics to fight it? the speaker realizes it's a virus, but are we killing good competitive bacteria?
So what we might be doing is what I mentioned about the trickler sands. So in the sanitizers, there may be compounds that are creating tolerance or resistance in the microbes, and therefore making it harder for the antibiotics to work. But the steer there would be to encourage people to use normal soap rather than ones with extra additives. And it's a bit of a mixed bag about whether we're using more antimicrobials or not, but it does look like they are being used more as well, you have to make a quick decision. You've got a very ill person, you don't really know what's going to make them better. The antimicrobial might do, but you're going to use a broad range one, which is going to then have an effect on lots of organisms, providing a selected advantage for the resistance, which is going to exacerbate the problem. Does that make sense?
So I have another question for you. To what extent are bioactive natural compounds, can they contribute to the control of AMR and to novel ways of finding multi-drug resistant bacteria?
Yeah, absolutely. I mean, that's part of the microbiology on Earth, getting everybody involved in trying to get natural environment samples so that we can perhaps find novel antimicrobials or maybe even find resistant mechanisms that we can study, understand, and try and find ways around. So absolutely, I think different environments are going to unearth different antimicrobials.
Okay, there's a question about new diagnostics underway this year. I'm not quite sure if you have any.
I don't know about any specifics. I just know that there's a big drive that we need to overcome, we need to take part in to get new diagnostics, because it's absolutely vital we know what is causing whatever the symptoms are and what the antimicrobial sensitivity profile of that organism is, so that we can use targeted antimicrobial therapy to safeguard the antimicrobials and to stop imposing a selection pressure.
Okay, last question. Azithromycin is used extensively beside the other drugs for COVID-19 patients during the pandemic. Are we expecting a new era of AMR to come?
Oh, I don't know, we could do. Certainly we might reduce the shelf life of the azithromycin, because if it gets used more and an existing resistance mechanism is more selective for and more.
Okay, thank you very much.
Environment, yeah. Sorry.
I think we will thank you for your time, and we'll move on to the next speaker. We may have time for more questions at the end. Okay, so our last speaker today is Dr. Oliver Elemento. He is director of the Englander Institute for Precision Medicine, an institute that uses genomics and informatics to make medicine more individualized. His team have developed a systematic approach to create tumor organoids across 12 tumor types and to use high throughput screening to identify optimal patient-specific anti-cancer treatment options. The focus of his research group is the systems biology of cancer. They focus on prostate cancer and hematological malignancies. Dr. Elemento is also Associate Program Director of the Clinical and Translational Science Center and Associate Director of the Institute for Computational Medicine. His talk is entitled The Future of Precision Medicine Towards a More Predictive Personalized Medicine. Dr. Elemento.
Thank you.
Your slides aren't up yet.
Yep, I'm sharing them right now. Here you go. Can you see them?
Yes, thank you.
Fantastic. All right, so it's a pleasure to be here today. I'm delighted to get a chance to tell you about our work in precision medicine, and more generally speaking about the field of precision medicine and how it's evolving, how it's changing. I'm going to start my talk by disclosures. Please, take a look at my disclosures and sort of get a sense of some of the potential conflicts. I think most of them are not really related to the talk today. I'd like to start my talks by giving a definition of precision medicine. Precision medicine is essentially a new form of personalized medicine that I believe is driven by technology. Technologies such as whole genome sequencing that allows us to sequence somebody's genome in just a few hours now, and in fact can actually allow sequencing of many genomes at the same time. But as you'll see in the second part of the talk, precision medicine is also more and more driven by data analytics and AI, and the combination of these technologies is really and really that will be the crux of the talk, is really making medicine more predictive. I also like to remind everyone that precision medicine relies on the availability of large cohorts as a way to learn how to connect, for example, genotypes and phenotypes. Using whole genome sequencing and sequencing in general, we can now sequence thousands, tens of thousands, maybe hundreds of thousands of individuals. And by gathering information about phenotypes, what disease they have, what disease they had in the past, we can make a connection, a robust connection between things that you measure in clinical samples, such as the DNA, and the occurrence of a particular phenotype. Once you've made those connections, then you can apply these connections to individual patients. But again, you really need to start with these large cohorts to be able to learn how to make these connections before you apply. So I'd like to say that precision medicine is learning from many and applying to one. Precision medicine is being applied to cancer. It's being applied to all kinds of disease, but from a more historical way, it's been applied to cancer maybe first before any other disease. The concept of precision medicine in cancer typically involves doing a biopsy, so getting cells from the tumor, also getting normal cells, for example, from the blood, and then sequencing the DNA of the tumor cells and sequencing the DNA of the normal cells from the same patient. The analysis of this DNA is focused on the identification of mutations, changes in the DNA that are specific to the tumor cells and not found in normal cells from the same individual. So that's what we do when we do this at Cornell. It's being done in pretty much all hospitals now in a lot of different countries. And it involves, as I said, DNA sequencing. As you'll see, it also more and more involves additional types of measurements on these tumor biopsies. One of them is, for example, the analysis of RNA, which is the genes that are expressed within the tumors. And I'll show you examples of how it helps in terms of understanding better the tumor and what's driving each individual cancer. This information is summarized into a report and then used as a way to make personalized recommendations for patients based on the DNA of the cancers. And so what we see when we sequence people's cancer genome is that every cancer is different. When two people get breast cancer, even though it is the same name, when you look at the mutations that drive the cancer in these two individuals, they are very often quite different from each other. And hence the importance of understanding through DNA sequencing what these tumors look like. So here is an example of a report that we use to convey the information of the tumor genome back to physician. So essentially, it's a list of mutations that we prioritize, sort of sort based on importance. We have the most important mutations at the top and less important mutations at the bottom. As you can see, most tumors that we sequence have quite a few mutations. So the challenge is to find these important mutations in the context of lots of maybe less important mutations. As I said, what we do more and more is to not only sequence the DNA, but also sequence the RNA. That allows us, for example, to credential certain mutations. This is a complex slide here, but it shows a report of a patient where we found a mutation in a gene called ERB2. We wanted to know whether this mutation was really active in this one patient, because not all mutations are active. They can be inherited from the past. As tumors evolve, they can inherit mutations. But what we did was to analyze the RNA of the same tumor where we found the mutation. And when we look for genes that are known to be downstream of this mutation, of a gene that has this mutation, we found that all those genes were actually quite highly expressed. So it gives us confidence that this gene called ERB2 here was actually really doing something in this tumor, because it was activating an entire expression program that's known to be downstream of this mutation. So the combination of these two modalities is very powerful in terms of credentialing mutations. This is an example here of what we can do once we find those mutations. This is a patient with a HER2 amplification. It's actually the same gene that I just showed you. This mutation was found to be amplified in this one patient. This is a patient here with bladder cancer. So typically, not a patient where you find this kind of mutation. You find this mutation often in breast cancer. But because this patient had very few options, our physicians decided to give that bladder cancer patient the breast cancer drug. It's called Herceptin. And as you see here in this patient, it had essentially a very sort of profound effect. It was able to completely eradicate tumors that had this mutation. So understanding what's driving individual cancers allows us to treat the cancers in a very personalized way as a result of being able to match it with specific drugs. Here's another patient with the same mutation, same breast cancer mutation, but here found in a uterine cancer, and also treated with Herceptin, the same drug that I showed you earlier, and likewise, also showing a very good response. So those two examples of patients are showing also a very sort of strong phenomenon in cancer now, which is that we're starting to move away from defining cancer based on where it's located in the body towards specifically what mutations are actually driving the cancer. And as a result of this kind of focus or change in focus, several organ-agnostic, biomarker-driven anti-cancer drugs have been approved by the FDA. These drugs have been shown to be effective in patients that have a specific mutation, no matter where the tumor is found in the body. It could be, a bladder tumor, it could be a colon tumor. These drugs tend to be effective, whatever the location, but if the mutation has a particular mutation, and there are several drugs like this now. So this is sort of where we are. This is where the field is at now in terms of genomics and utilization of DNA sequencing as well as RNA sequencing, as I showed you. What I want to do in the next few slides is to give you a sense of where the field is going. As I said, my belief is that the field is moving towards a more predictive form of personalized medicine, and that actually very much involves the second part of this figure that I showed earlier, which is the utilization of AI and data analytics. So this is something that we've been doing here at Cornell in my group for some time. A few years ago, we showed that using a form of AI called support vector machine, we could diagnose very precisely whether a thyroid lesion, basically a lesion that you find in the thyroid in some individuals, which tend to be pretty common. Some of these lesions are malignant, some of them are benign. We showed that using the analysis of microRNAs, which are tiny genes that you find in tumor cells as well as normal cells, combined using a machine learning approach, as I said, called support vector machine, you could actually achieve a diagnosis accuracy of about 90%. So this was kind of our first foray into the field of AI and machine learning applied to precision medicine. And since then, we've actually applied this to many different problems. One of them was to, for example, predict which patients are going to respond to immunotherapy. Immunotherapy, as you know, is the ability to revive the immune system to target cancer cells, a very promising form of therapy for cancer, but unfortunately, it does not work in many cancer patients. So the challenge in the field has been to find the patients who are likely responders to immune therapy. What we showed in this paper, and this is something we're continuing to do research about, is that the integration of data that you get from patients and the biopsy results for the DNA sequencing, the RNA sequencing, the integration of these data using a machine learning method, also called a random forest in this particular case, was actually able to build a predictor that can predict responders with about close to 90% accuracy. And we show that it's only by combining the different signals that you get from throughout the genome and the genes that are expressed that you can actually reach this kind of performance in terms of prediction. So again, this is the future of sort of precision medicine is really with data integration for multiple signals that give you a full picture about the likelihood of response to particular drugs of certain patients. We're moving away from using one marker at a time, using a combination of markers combined and integrated using machine learning and AI. Machine learning is used now in the form of deep neural networks very much as a way to not only analyze genomic data, DNA data and RNA data, but also analyze medical imaging data. This is exciting to me because this is a source of data that's readily available there's a tremendous amount of medical data that's in the medical records of, sort of pretty much all patients, for example, patients who get cancer. And, we think there is an opportunity to combine this data with maybe genomics data as a way to deliver better, more precise precision medicine. So again, this kind of AI is really completely driven by the availability of these deep neural networks, which very often are pre-trained. What it means by pre-training is that they are pre-trained on lots of different images, so they're pre-trained to understand, certain fundamental concepts about images, such as, textures, objects, and so on. And what we do is to retrain them on medical images, and we don't necessarily need a lot of medical images to do so, just because we have already pre-trained those networks on large data sets. So these data sets are being used to analyze all kinds of medical imaging data. And these are things that we do also in my group and then with my collaborators here at Cornell. We apply this to histopathology, which is the analysis of tissue to enable diagnosis. We apply this to radiology, which is, as you can see here, it's analysis of organs at a resolution that allows you to find, for example, certain tumors. One thing that we did a few years ago was to demonstrate that deep learning applied to histology, which is again tissue sections from cancer, for example, stained in a way to reveal cancer cells and the morphology of cancer cells. AI can actually be used and trained on this kind of images to distinguish between two types of lung cancer. This was one of the first applications of AI to histology, demonstrating that you can essentially achieve extremely good accuracy in terms of distinction between two types of cancers that are actually quite similar to each other. But AI is able to pick up signals that can distinguish these types of cancers. We showed then we can apply the same kind of AI to breast cancer to identify different subtypes of breast cancer based, again, on H&E, based on histopathology slides. We also showed that a few years ago, working with a company, that you can use AI, the same kind of deep neural networks, as a way to predict prognosis, for example, in mesothelioma, better again than everything that have been sort of done before, again by integrating data from these images, and we had a lot of them in this particular case. One exciting application of AI is the ability to connect different types of imaging, for example, radiology and histology. One thing that we used AI for was the application of AI to prostate cancer in this particular case. We're trying to show that we can predict the result of a biopsy of a prostate for somebody with prostate cancer just based on radiology images. We had a large data set where we had both radiology images and histology with a Gleason score and an assessment of the aggressive nature of the cancer. And we showed that using AI, we can actually map one into the other. What it shows is that essentially we can move towards a future where we don't necessarily need to do a biopsy, because we learned how to train an AI to analyze the radiology images to basically reproduce the result of a biopsy. So that's very exciting and definitely ongoing work that we're trying to expand to other diseases as well. And these applications of imaging not only focus on cancer. For example, we've also showed last year that you can use the same kind of neural network as a way to analyze different types of medical imaging data. For example, data from an IVF treatment center here at Cornell. We had lots of embryos that were fertilized, human embryos, images that you see here. Some of them were going to be implanted and would give rise to a live birth after IVF. Some of them wouldn't do so. We were able to train an algorithm to be able to distinguish good embryos that are going to be successful versus poor embryos that are not going to be successful. Again, pointing to the idea that you can essentially kind of tack on AI to this kind of medical imaging as a way to personalize the analysis of these images and improve, hopefully, the outcomes using this kind of data. So this is sort of basically the end of the talk. What I wanted to give you a sense, I only had a few minutes, but I'm hoping I was able to give you a sense that multi-scale data integration, genomics, imaging, for example, combined and integrated using AI is making precision medicine more predictive. And obviously this is a great thing for patients. This means more personalized medicine for everyone. And this is we think that this is very exciting. So I'll stop here and I'll be happy to take questions.
Thank you very much for your truly captivating presentation, showing us how precision medicine could, or actually will, affect or is affecting our healthcare future. So first question I have for you here is, Will informaticians become our future medical diagnosticians? Perhaps this would be a career opportunity for consideration.
Yeah, that's a wonderful question. I don't think that's going to be the case because obviously a pathologist and, like you said, diagnostician in general obviously have a lot of medical expertise that they use as a way to not only analyze this kind of data, but also sort of communicate information to the patients and so on, and obviously integrate lots of decades of education in order to combine certain type of data with other things that they know about the patient. But I do think that informatics is going to be more integrated into the medical system in general, make the medical system more predictive, and I do think that more and more diagnosticians are going to rely on these kind of systems as a way to be more productive and be more performant. So I think this is a great part of the future. I think that again, the skills, building skill sets in terms of informatics and being able to interact better with machines and understand the result of an AI prediction, is going to be critical in terms of, for example, medical education.
I have a question here. How likely is this technique to give false positives?
Right. So, I mean, some of these techniques, depending on the sort of, the performance of the algorithms, indeed do give false positives and false negatives. But, just keep in mind that everything that we do now in the medical system also does. So I think there's, multiple sort of studies that have shown that, for example, in the diagnosis of melanomas, from images of moles, this kind of AI applied to thousands and thousands of images gives rise to to performances that are comparable, if not better, than what humans sort of could do. And and humans are not by no means perfect at making these diagnoses. Everybody makes mistakes from time to time, and this is absolutely part of the issue. We think that AI has a possibility to interact, basically collaborate with human beings. That's a way to make the human beings more performant.
So related to that, I have a question. Is computational power becoming a bottleneck?
Yeah, so I mean, it is because our ability to generate data is really now, very, very sort of substantial. And imaging data actually requires specific types of computational infrastructure called GPUs, these graphical processor units. So we actually do see bottlenecks in terms of being able to train AI models for analyzing images because we don't necessarily always have enough access to these kind of computers. It is a tractable problem in some ways, but it is an important problem. And to be able to sort of deploy AI at scale and learn from extremely large data sets, more and more you need access to very large computing power that is not always available to everyone.
Another question for you is there any way you could make precision medicine more accessible for third world countries?
Yeah, and that's a wonderful question, and I absolutely completely embrace this idea. I think that a lot of the precision medicine that exists now is not broadly accessible. Even within the US, for example, there are centers that are just not equipped, that haven't had a chance to invest in the same kind of, for example, even sequencing type of analysis that I showed at the beginning. So I think there's a need to decrease the cost of these assays. They are still expensive for a variety of different reasons. They're not accessible enough. So I think there is a very strong need to decrease the cost and make this kind of medicine more accessible. It is the future of medicine in general, but we need to make sure that it's accessible to everyone, and absolutely indeed to other countries that don't necessarily have right now the infrastructure that we have here in a more modern country.
Right. I would like to thank you very much, and I think owing to the time now, I'd like to thank you, Dr. Elemento, Drs. Frontzek and Hardie, for the time they've spent sharing their research and all of the thought-provoking presentations. Before I end the meeting, I would like to close with a few announcements. First, I'd like to thank everyone for attending the webinar and the speakers for their presentations. You can continue the conversation online. Follow @BiochemSoc and @PP Publishing on Twitter. We welcome suggestions for future topics and speakers to feature in this Biochemistry Focus webinar series. We invite you to submit a proposal if you have an idea for an upcoming webinar. You can find more information about the webinars and propose your webinar on www.biochemistry.org/webinars. Join us for the next webinar in the series entitled Open Access, Ask Me Anything, on Wednesday, the 14th of October at 1500 BST, ahead of Open Access Week. Ask your questions and have them answered by a panel able to share perspectives from an institution, researchers at different career stages, and a publisher. In these new and challenging times, it's more important than ever to stay connected. It's an extraordinary time for all of us, but it's also an exciting time to join the Biochemical Society as they look to expand their online activities. Join their community of researchers and specialists to stay connected and take advantage of key benefits, including discounted registration fees for society courses and meetings, exclusive access to a wide range of grants and bursaries, personal online access, and 2 other journals and more. Visit their website and more. And so in closing, thank you to all the speakers.
Thank you.
Thank you.
Thank you.